Good morning, all. We're going to pick up where we left off, which was talking about the cerebrum, and more specifically, the cerebral cortex. I think the most important thing to keep in mind when talking about the cortex is that this is gray matter, so predominantly inner neurons, cell bodies, their dendrites with their unmyelinated axons. And the cerebral cortex is actually made up of billions of these inner neurons and makes up about 40% of total brain mass. And what we've been able to do over time is actually kind of map out 47 structurally distinct areas along the cerebral cortex. We refer to these as Brodmann areas. And even furthermore, we've been able to kind of categorize these based on their function. So we do have different functional area categories. We have sensory areas. These are going to be areas of cortical tissue that make us consciously aware of a particular sensory stimulus. We have association areas, which are going to be cortical areas that kind of further process and integrate sensory information. It can be just a particular stimulus, like with a sensory association area, or it can be multiple sensory inputs that are being further integrated and processed in our multimodal association areas. And then last but not least, we have a motor area category. This is going to be cortical tissue that are responsible for dictating motor outputs or planning varying motor functions. So all of our voluntary motor functions are controlled by these motor areas within the cortical tissue. So if we look at the image here, we'll notice that we kind of have things color coordinated. So we highlight our primary motor cortex, we highlight different motor association cortical areas in a different color. We see all of that kind of within that frontal lobe. We see our primary sensory cortex and then sensory association cortexes, and are located more into that parietal, occipital, and temporal lobes, and then our multimodal association areas that kind of spanned again through the parietal and the temporal as well as into the frontal lobe. So we're going to talk first through the different sensory areas, and the hopes is that you kind of start to put together like this bigger picture where we first take in sensory information and we're made aware of it. And then from that point, we give that sensory information some meaning. What is it? Where are we receiving it from? And then we take in all of the sensory information from our entire external and internal environment, and we use that information to dictate the most appropriate motor response. In order for all that to start and to happen, we have to be able to be made aware of our different sensory stimuli. So within our sensory areas, we break those down into primary sensory cortical areas and sensory association areas, where the primary sensory cortex is just making us aware of a sensory stimulus, and the sensory association area is actually doing further interpretation, integration of that information, and giving meaning to that particular sensation. So we're going to talk through kind of the different primary sensory cortex areas, as well as talk about any of the sensory association areas that might be present in regards to those different sensations. What we need to make sure that we are able to do is break down kind of our general senses versus our special senses, and then appreciate what somatic versus visceral means. So when we think about our primary somatosensory cortex, this is going to be cortical tissue that is responsible for receiving, sensory input from a number of general somatic senses. So somatic meaning skin, muscle, bone, and general meaning things like touch, pressure, stretch, pain. Those are all going to arrive in a particular area along this primary somatosensory cortex, which we find more specifically in the postcentral gyrus of the parietal lobe. And you can see that highlighted there in a more royal blue color. Now, what's really interesting about this postcentral gyrus and the primary somatosensory cortex is that it has what we call spatial discrimination, meaning that we've been able to map along the postcentral gyrus specific cortical tissue responsible for specific areas of the body. This allows us to precisely locate where a stimulus is presenting itself at. So this map that we have is referred to as a sensory homunculus. I always get caught up on that word. And we can see it here mapped out, right? We'll look at it here on the right, and we have different areas of the body kind of lying along that postcentral gyrus. And what that allows us to see is that areas that are exposed, right, to more sensory input, or areas that are more sensitive, if you will, are going to have more cortical tissue responsible for receiving sensory input. So if we think about the hands, for example, that are always touching, always manipulating objects, we would expect there to be a great amount of cortical tissue responsible for receiving sensory information from the hand. We'd expect the same thing along the face and along the mouth. And we would expect the same thing kind of along the foot, right? Any area that is constantly receiving touch, pressure, stretch, sensation, we think about even the tongue and the chemical, right, concentrations that change. All that sensory input will make its way, right, up to the somatosensory cortex, right, in that postcentral gyrus, so that we can be made aware of that sensation. Something to cue into is that the projection is contralateral, meaning that something that I might be manipulating in my right hand, right, I'm going to receive information about that touch and about that pressure in my left primary somatosensory cortex. Okay. So then just posterior to the primary somatosensory cortex is where we will find our somatosensory association area. Okay. Your somatosensory association area is going to be cortical tissue that is responsible for integrating the sensory input that was received by the primary somatosensory cortex. So actually giving this a little bit more meaning. Okay? We already know the where in this situation because of that spatial discrimination, but we don't necessarily know the what. So we need to be able to pull from other stored memories, right, from other strong synapses to kind of generate more of the what in regards to this sensory stimulus. And again, you can see that that's located in the parietal lobe. So then when we think about our special senses, such as vision, taste, right, smell, those are all going to have their own primary cortical areas. And then some of them will also have association sensory areas to help further process some of their information. Okay? So your primary visual cortex is going to be located in the occipital lobe. We can see it right here on the most kind of posterior inferior aspect of the occipital lobe. And the job of this cortical tissue is really to just receive the visual information from the retina and make us aware that visual input, that light, right, we are being exposed to light. But at this time, we don't really know the what and we don't know the where. We're not giving any color, any form, anything to whatever image we're being exposed to. Right? It's not until, right, information is sent to our visual association area, which we also find here in the occipital lobe, kind of around the primary visual cortex, right? And this visual association area is what actually integrates and processes all of that visual stimuli and creates the form, the shape, right? Gives it color, right? Allows us to tell whether the image is static or in movement, dynamic. So we start to generate a little bit more of the what and where once synapses happen from the primary visual cortex to the visual association area. When we have our primary auditory cortex, so our ability to be made aware of a sound, right? Ability to have kind of that just general conscious awareness of any sound stimuli has to do with synapses arriving at the primary auditory cortex. Primary auditory cortex is located in the temporal lobe. And we can kind of see that guy right here, kind of right at that junction along the lateral sulcus, right? And again, just making us aware of sound sensation, but not allowing us to really understand necessarily where the sound is coming from or what the sound actually is. That's going to be carried out by the association area. When we think about the association area, it's going to be here in blue, right? And I just circled it with the red pen there. And that's where the where and the what start to come into play. And so, again, the association area is all about further processing and integrating. And it may be receiving, right, information from other areas of the brain, some of those multimodal association areas, to further enhance that what and where. And you also notice that part of the auditory association area kind of creeps into Wernicke's area, which we'll talk about a little bit, how that plays a role in language comprehension. When we think about our sense of smell or olfaction, right, our ability to become aware of the smell sensation is based on information received by the olfactory cortex. We find the olfactory cortex located on the temporal lobe, but even more specifically on the piriform cortex, deep on the uncus, which is just a small portion, right, kind of a subdivision of the temporal lobe. So we can see that here kind of highlighted in pink, right? And when we think about the uncus, a lot of times people refer to that as being a region of the rhinencephalon, which plays a role in the limbic system, which is a functional brain system that's responsible for kind of a lot of our processing of emotions and eliciting emotional responses. So it makes sense that the olfactory cortex is kind of found in this area if you take into consideration how certain smells can elicit different emotional responses. Located in the insula, which we talked about being kind of that fifth lobe of the cerebrum that's found deep, right? Deep into that lateral sulcus. You have to pry away the frontal lobe from the temporal lobe in order to really latch on and see the insula. But that's where we're going to find our gustatory cortex. Your gustatory cortex is going to be cortical tissue that makes us aware of a particular taste stimuli. We can see that highlighted right here in purple. And notice that neither of these have their own specific sensory association areas, but do realize that information from these primary cortex areas is going to head to multimodal areas and all be interpreted and processed as part of a bigger planning event. So if we stay on that insula, right, and in this image we've actually removed a big chunk of frontal, parietal, and temporal in order to expose kind of the cortical tissue of the insula. We'll find both our vestibular cortex and our visceral sensory cortex. So with your vestibular cortex, you're going to find that in the anterior lobule of the insula. So everything here in blue, all that cortical tissue would be housing neurons that is going to receive information that makes us aware of our equilibrium. And when you think about equilibrium, think more about kind of our head position and where our head is at in space. And then even further beyond that, again, whether it's static kind of versus dynamic, whether we have any linear acceleration or we're at a standstill. Then we think about the visceral sensory cortex, that's going to be kind of on the cortical tissue here of the posterior lobule, and that's going to make us aware of general senses that are inputs traveling from the thoracic and abdominal organs. So we think again about what might be happening in those organs as far as stretch, right, or chemical change. That's all we're going to be able to appreciate that is actually taking place when we receive that information at the visceral sensory cortex in the insula. Okay, so if we understand our sensory areas, we can then transition into our multimodal association areas. We have three multimodal association areas: posterior association area, anterior association area, which is also referred to as the prefrontal cortex, and then the limbic association area. Your posterior association area is going to be located in the parietal and temporal lobes, and this kind of makes sense if you think about what it's receiving, and what it's going to be receiving is information from the visual, the auditory, and the somatosensory association areas. Okay, so all of this flood, right, of sensory input is going to present itself to this particular cortical tissue in the posterior association area so that we can further process kind of the what and the where, especially in regards to visual and auditory stimulus, right? And then give us this overall sense of where we're at in space. So kind of the spatial orientation or a body sense. So it just means that we take into account all of our external environment, right, and some of our internal environment in order to make sure that we can understand and appreciate where we're at in space before we go ahead and initiate any motor plans. Okay? In addition to it processing, right, further visual, auditory, and somatosensory stimuli, it is also an area in which we find Wernicke's area, which is a specific area of cortical tissue that's responsible for language comprehension. So our ability to give meaning to what we hear during conversation predominantly. So, and that makes sense again if you think about what else is being processed in this area as far as the auditory and visual information. So, visual information, us actually watching an individual's mouth move, plays a role in comprehension, and then hearing what a person is saying plays a role in that comprehension as well. Then we have our prefrontal cortex, also known as our anterior association area. It's going to process information from the two other multimodal association areas, so the posterior association area and the limbic association area, and it's going to use that information to kind of help plan and initiate some motor responses. It will then share that with the premotor cortex, who will then, right, talk with other areas within the brain before getting that information to the primary motor cortex. In addition, it plays a substantial role in cognitive function. So when we think about our intellectual abilities, our abilities to think critically, to problem solve, that has everything to do with us taking into account a number of sensory stimuli as well as feeding off of memories in order to carry out those cognitive functions. And then the limbic association area is just that. It's kind of the emotional side of things, and it plays a big role in the memory piece. So a lot of times the motor responses that are elicited are done so based on how we've responded in the past to particular events, right? So that kind of whole idea of motor memory, of emotional memory, the limbic association area plays a big role in that. And so this is, again, just talking about the cerebral cortex that plays a role in that. It's not talking about the limbic system as a whole, which spans across the entire brain. So then from here we talk about our motor areas, and what's kind of nice about our motor areas is that they're all located in the frontal lobe. So we kind of start with our primary motor cortex here, move back into our premotor cortex here, then transition back into our frontal eye field, and then down into our Broca's area. Okay? All of this cortical tissue is responsible for processing and generating motor output, okay? And then eventually we send that motor output out to the periphery in order for our skeletal muscles to contract. So we'll start with our primary motor cortex, which is really who controls the execution of all of our movements, all of our voluntary movements. So it's not necessarily controlling the movements as far as planning them or coordinating them, sequencing them, but it is who is actually going to dictate the motor output. So when we're ready to perform a movement, that electrical impulse is going to leave from the primary motor cortex. And we find that in the precentral gyrus, in that frontal lobe. And we've heard this word before, pyramidal, right? Motor tracts, we talked about how they travel down through the brainstem. Well, of course, they arise as pyramidal neurons, so the tract part is just their axons, but their cell bodies and their dendrites are what make up the primary motor cortex. Okay? And we can map out areas along the precentral gyrus and how they correspond to different areas of the body, again, via a motor homunculus, right? And this motor homunculus is going to be kind of specific neurons responsible for controlling specific areas of the body, and we should be able to see that areas that are in constant movement, right, such as our hands always manipulating things, all that fine, right, motor processing that we have going on with the hands, all going to be carried out by this primary motor cortex and these pyramidal neurons. And so we would expect on the motor homunculus for the hand to kind of take up a larger chunk of the cortical tissue. And so if we look at the map here, that's kind of exactly what we see. Going to look like all this cortical tissue is going to be responsible for sending motor output to the hands and fingers. We see that how much it takes in sensory-wise is pretty similar as far as the gross amount of tissue. Same thing like with the face and the mouth, right? We're constantly activating muscles of the face for facial expressions. We're constantly moving our mouth. So when we're talking, that's all generating voluntary movements that's been sent out from the motor cortex. The premotor cortex that's located, again, in that frontal lobe just anterior to the precentral gyrus does a lot more of planning of movements. So when we think about more of our complex movements, when we think about multi-joint movements and multi-planar movements, the more complex the movement, the more involved the premotor cortex is going to be. It's going to be in constant communication with other areas of the brain to help kind of plan these movements. And the reason that we have this kind of constant communication, again, is just to make sure that whatever movement we dictate, whatever response we send out to the skeletal muscle is based on what we're receiving sensory information-wise in that moment. So whatever we're being exposed to in the environment in that moment is going to dictate how the premotor cortex plans and what information it then sends to the primary motor cortex to then shoot out to the periphery. Your frontal eye field is a small area of cortical tissue that's responsible for just voluntary eye movement. So it's going to send out the motor output to our extrinsic eye muscles so that our eyeballs can move as we track a moving object or as we stabilize, right? As we stabilize our gaze on a particular object and or objects. Broca's area is again a very small cortical area with a very specific function in regards to motor output to muscles that we need in order to carry out speech. Okay, so it controls all of our necessary movements for speaking. It's located only in the left hemisphere, and it's going to be communicating back and forth with the posterior association area with that Wernicke's area that was responsible for language comprehension. And so as we are taking in auditory information, especially in conversation, and we're comprehending what a person says, and then we're taking in their body language as well via different synapses in the posterior association area, we then send that information to Broca's area so that it sends out the right motor output to muscles so that we generate the appropriate response from a speaking standpoint. So again, when you think about the cerebral cortex, it's all about receiving, processing, integrating information in these inner neurons, right? From there, we know that different areas need to be, of the brain, especially of that cortical tissue, need to be able to talk, right, with each other in order to make sure that what we're processing and what we're planning is all coincides with our particular environment in the moment. That's where the need for white matter comes into play. So we're going to find cerebral white matter deep to the cortex. This is going to be primarily myelinated axons, but unmyelinated axons will be present as well. And they're all going to be bundled into what we call tracts. Okay, so you think about the central nervous system. Bundles of axons in the central nervous system are tracts. Bundles of axons outside the central nervous system are nerves. Okay, so tracts and nerves are similar from a structural standpoint, but not from a location standpoint. Okay. So when we think about the tracts allowing for this cerebral cortex communication, it's then trying to figure out who they're allowing for communication with, right? So we have cortex to cortex, and that can be same hemisphere or it can be opposing hemispheres. We have cortex to the brainstem, cortex to the spinal cord. And so we have three different types of tracts that can present. We have association fibers that you'll see kind of depicted here in red. Okay, and these are going to allow for cortex to cortex communication within the same hemisphere. So if we think about, for example, how that visual association area wants to be in constant communication with the posterior association area, we need to be able to send information from different cortical areas in order to further process, integrate, and give meaning to that sensory stimulus. Okay. So our association fibers, same hemisphere, cortex to cortex. We then have commissures. Commissures are what allow our right and left hemispheres to kind of communicate with each other. So we'll allow for long axons to travel across from hemisphere to hemisphere. The largest of the commissures on the brain is the corpus callosum, and that's depicted there in green. We then have projection fibers. Projection fibers are going to do just as their name would suggest, right? They're going to transmit or they're going to project electrical impulses kind of either down from the cerebral cortex to the lower levels or from lower levels of the central nervous system up to the cerebral cortex. We can see those highlighted in purple, although I want you to cue into kind of hear how we see some fibers traveling to and from the thalamus to the cortex, right? So that's that whole idea that we have constant arousal of the cortex via the thalamus, or how that thalamus acts as a gateway. We also notice that we see some fibers that are projecting down and then crossing over at the decussation of the pyramids in the medulla. So those fibers will be representative of our pyramidal motor tracts, right? So the pyramidal neurons actually being in the primary motor cortex, and that being their cell bodies and dendrites, and then their axons actually traveling, right, the entire length of the cerebrum through the brainstem, crossing over, and then into the spinal cord, and eventually out into the periphery. Okay, so your projection fibers, again, are allowing for that communication in both directions. The example shown here, these would be motor fibers. So then in addition to the gray matter in the cerebral cortex, we also have gray matter that's kind of located deep within the cerebrum. So you got outer gray, you got internal white, and then you have internal or deep gray matter, right? And one of the first kind of nuclei, right, that present themselves as deep gray matter are the basal nuclei. We also see them in text as the basal ganglia. Your basal ganglia are made up of kind of four distinct brain nuclei: the claustrum, the caudate nucleus, the putamen, and the globus pallidus. And all of these guys are in communication with each other, as well as in communication with higher regions of the brain, playing a really important role in kind of the initiating of a lot of our voluntary movements, the halting, right, of some of our voluntary movements, and then regulating intensity. So remember when we talked about the midbrain, we talked about the substantia nigra, talked about degeneration of the substantia nigra leading to a decline in dopamine, which can result in Parkinson's disease. So part of that has a lot to do with the basal ganglia as well, because what that dopamine does and how the midbrain communicates with the basal ganglia is via dopamine as a neurotransmitter, is it plays a role in inhibiting the basal ganglia, right? And we need that constant or appropriate inhibition to make sure that the basal ganglia isn't kind of overstepping its bounds, and that it's always kind of doing more of a appropriate measure of starting, stopping, and regulating movement intensity. So when you lack that inhibition, that's when you can get those rapid, jerky movements, or that's where you can get that gait freeze that you oftentimes see with individuals with Parkinson's. Another area of deep gray matter is the basal forebrain nuclei. Your basal forebrain nuclei is part of what we call the cholinergic system. And these are brain structures that are responsible for producing and secreting acetylcholine. Acetylcholine is an excitatory neurotransmitter, at least how we're using it in terms of the function of the basal forebrain nuclei. And so its ability to produce and secrete acetylcholine and then to contribute to the synapses of the prefrontal cortex, as well as its communication with the thalamus, then plays a really big role in cortical arousal, and it plays a really important role in our ability to learn and form memories. And then in regards to motor control, that has a lot to do with its communication with the prefrontal cortex and how the prefrontal cortex then communicates with the premotor cortex. You can see that there's a lot of communication with these basal forebrain nuclei via, projection fibers then to the varying cortical areas of the frontal lobe. And then last but not least, as far as deep gray matter, is the amygdaloid body. The amygdaloid body is a cerebral structure that contributes to the limbic system. So again, the limbic system is a functional brain system, meaning it is areas within the brain, spanned, widely distributed throughout the brain, that then are all in communication with each other in regards to processing emotion, making us aware of varying stimuli that induce emotional responses. And when we think about the amygdaloid body, it plays the primary role in processing fear and then stimulating appropriate responses to fear. Okay, so now that we can kind of understand the brain, break it down into its regions, break those regions down into subregions, and then even further than that, break it down into different tissue areas, whether that be cortical areas, whether it be white matter areas, whether it be brain nuclei, we understand that the brain is in constant communication, throughout the entirety of it, So widely distributed areas within the brain are always talking to make sure that we can dictate the most appropriate motor response based on our external and internal environments. So if we understand that, we can understand how any form of inhibition or dysfunction pathology to one area of the brain will affect all areas of the brain. So of course the body has built-in protective mechanisms, to make sure that the brain, stays away from harm, whether that is something that could present to the brain internally, or if that's something that could present to the brain externally, as far as trauma. So first and foremost, the brain is completely encased and protected by the skull, and that again is to try to minimize exposure to the brain, but then also to protect the brain from external trauma. Meninges, which is connective tissue layers, completely encase the brain, and we'll talk about kind of their role in protective mechanism as far as kind of keeping things out from the exterior, but then also providing the brain with appropriate nutrients by protecting the blood vessels that penetrate the brain. Cerebral spinal fluid plays an important role in brain protection, and then the blood-brain barrier. So when we think about the meninges, again, the meninges are layers of connective tissue that completely cover, encase the central nervous system, so both the brain and the spinal cord. Their role is to protect the brain and spinal cord from the exterior, as well as to protect and enclose the blood vessels that supply it. They also contain cerebral spinal fluid, which plays an important role in providing nutrients to the brain. Meninges include 3 layers: the dura mater, the arachnoid mater, and the pia mater. The dura mater is the outermost, strongest layer. Its name literally means tough mother, and it is a 2-layered structure So the first layer is the periosteal layer, and that's going to be found right on the inner surface of the bone. So if we look here, start from the exterior, we have the scalp, we have the periosteum, which covers the bone, we have the bone itself, right? So we've got compact bone, diploid, compact bone, and then on that underlying surface of the bone, we have the periosteal layer of the dura mater. And this is going to be dense connective tissue, right? And then as we go inside from that, we have the true covering of the brain, also form a dense connective tissue, and that is going to be the meningeal layer. Okay. In addition to like just creating this full encompassing around the brain and providing kind of that exterior border, it also encloses the dural sinuses, which you guys know are the venous return from the brain. And you can see here the superior sagittal sinus, how it is being enclosed between the two layers, between the periosteal and the meningeal layer. Okay. The dura mater goes a step further as far as not just completely wrapping in the brain, but then also sending in partitions that kind of separate different regions from each other while also housing those dural sinuses and allowing for appropriate venous return, as well as just creating a little bit stronger resiliency of the meninges in support of the brain. So we can see that we have a partition here of dura mater that kind of invades the longitudinal fissure, so resting between our right and left cerebral hemispheres is the falx cerebri. And we can see that components of the falx cerebri will encase the superior sagittal sinus as well as the inferior sagittal sinus. Okay. We then see partition here, and we see it predominantly just on the right because it's been cut away on the left, but that's going to be a partition of dura that rests in the lateral fissure, or excuse me, the transverse fissure, so in between the cerebrum and the cerebellum. And this is the tentorium cerebelli. The name means tent over the cerebellum. Okay. And then we see a small partition here that would be resting between the two cerebellar hemispheres, the right and left cerebellar hemispheres, literally laying right on the exterior of the vermis, and that is the falx cerebelli. So as we travel in deeper from the dura mater, we get to the arachnoid mater, and arachnoid referring to spider, right? Really because of the presentation of the arachnoid mater looking kind of like a spider web. That's where it gets its name. Prior to hitting the arachnoid mater, we do have a potential space between the dura and arachnoid mater referred to as the subdural space. We say potential because nothing should really be in this space, but it could be a site for fluid accumulation such as blood, like if you had trauma to the head causes a bleed, that blood could present itself in this subdural space, creating a substantial amount of pressure. It could also be a potential space for infection, which is really dangerous because if it is able then to get into the arachnoid space, or the subarachnoid space, so penetrate through the arachnoid mater, that's where we're going to find cerebrospinal fluid. So that could be really problematic, especially if we think about that getting into the brain tissues itself, or if even finding a way into the blood vessels that are supplied by the brain that are also present in that subarachnoid space. Okay? So then between the arachnoid mater and the pia mater, that's where you find that subarachnoid space. Again, it's filled with cerebrospinal fluid. It's predominantly loose connective tissue, so that's what gives it more of a spider web type appearance. It's an area in which you'll find a lot of the blood vessels that supply the brain before they then branch into smaller vessels and travel inward with the pia mater. Also unique to the arachnoid mater is the presence of arachnoid villi, which we can see right here. And you'll also see these in text as arachnoid granulations. We can see these in lab. So if you look either on the undersurface of the dura mater that we've taken out, or on the surface of the brain itself, if the arachnoid mater hasn't been too interrupted, you can see remnants of these arachnoid villi. And they actually allow for a passage of cerebral spinal fluid into the dural sinuses, meaning that we are constantly recycling our cerebral spinal fluid. So then as we travel deep, we get to our thinnest layer and our deepest layer, that is the pia mater. Its name means fine mother, so it's a very delicate connective tissue that clings very tightly to the surface of the brain, oftentimes pulling in, right, small blood vessels that can then penetrate into the outer surfaces of the brain to supply nutrients. So then you've already seen this name a couple of times as we talked about the meninges, but it's cerebral spinal fluid, or CSF. CSF is a blood filtrate, or more specifically a plasma filtrate, and it plays a really important role in nourishing the brain. So a lot of the components of your cerebral spinal fluid are similar to that of blood plasma, and we know that the role of blood plasma is to nourish all of the cells and tissues of the body, so it makes sense that that's kind of the same when we talk about the brain and the spinal cord. The cerebral spinal fluid is formed by choroid plexus. So choroid plexus is kind of a twofold structure. It's first of a network of capillaries that are then lined with ependymal cells. And these ependymal cells act as little filtering cells. So as plasma leaves the capillary, right, through those intercellular clefts, the content will then travel through the ependymal cells, where the ependymal cells can kind of act a bit as a filtering device, making sure that whatever then enters into the ventricles, right, the actual components of cerebral spinal fluid, are all good stuff. So we're thinking about glucose, of course, oxygen, varying vitamins, lots of different ions. So when we think about what we need in order for our synapses to take place, right, we need calcium, we need chloride, we need sodium, we need magnesium, we need potassium. Those are all essential to carry out, right, synapses. So in addition to like providing that nutrients through the formation of this blood filtrate, and the kind of on a grosser scale, cerebral spinal fluid also acts to kind of cushion the brain and the spinal cord. So it's this liquid cushion that creates some buoyancy, right? So then as we're moving around, because all day long we're moving and our head is turning up and back and forth and up and down, and we have little minor traumas, someone pats you on the back and your head kind of jets forward a little bit, meaning the brain is moving, but you have something in there to allow for a little bit of buoyancy and to take on some of that force. Okay. So now what we got to think about is kind of the big picture. Like if we understand the kind of how the cavities present themselves, like the ventricles, and how they kind of allow for flow, and we understand how now the meninges, that subarachnoid space, then allows for flow of cerebral spinal fluid, and then allows for cerebral spinal fluid to enter the dural sinuses, we should be able to create kind of a full flow pattern with circulation of cerebral spinal fluid back into venous return. So that's what we're going to do now on this image. We're going to start up in our lateral ventricles, which are indicated by the letter 1 here. You can see the choroid plexus hanging out in those lateral ventricles. So we're going to have cerebral spinal fluid being produced here. Cerebral spinal fluid will then travel from a lateral ventricle through an interventricular foramen into the third ventricle. We know the third ventricle is encased within the diencephalon, within the thalamus and hypothalamus. And then from our third ventricle, we will have fluid travel through the cerebral aqueduct into the fourth ventricle. Now, once it reaches the fourth ventricle, it has some options. Remember, it can either go through the lateral aperture, so we have 2 lateral apertures, or through a single median aperture, or it continues on through the central canal of the medulla and into the spinal cord. Okay? So if it chooses to go through an aperture, right, those openings open up to the subarachnoid space, meaning from the fourth ventricle, we can have flow of our cerebral spinal fluid into the subdural space. So here we got flow through the median aperture, and here we got throat flow potentially through a lateral aperture, But what this then allows for is now we're in the subarachnoid space, and we can travel through that subarachnoid space, right, all the way up, find ourselves in a present the fluid to a granulation, which then allows that cerebrospinal fluid to enter into a dural sinus. And I know this image just depicts those granulations kind of along the superior aspect, but we'll see those granulations along most of our dural sinuses. So not just superior sagittal, but you'll also see those granulations present along the transverse sagittal and the straight, the transverse sinus and the straight sinus as well. But what that allows us to appreciate is that cerebral spinal fluid is constantly being recycled. So we allow all that content and potential waste, right, to enter into the dural sinuses, where it can then go through circulation back to the heart and through pulmonary circulation and back out. And at the same time that we're recycling it into venous return, right, our capillaries in our ventricles are creating more cerebral spinal fluid, more of that plasma filtrate. Okay. So then last but not least, as far as built-in protective mechanisms for the brain, is the blood-brain barrier, right? And when we think about blood-brain barrier, we think about just actually some structurally adapted capillaries that try to negate, right, varying solutes from entering into the brain. So the idea here is that we can prevent like any potential toxins from getting in the brain and inhibiting, function of all of those very, very precious brain regions that we've talked about. So the blood-brain barrier are just specialized low permeability capillaries. Okay, and so when we think about a continuous capillary, which is the most common capillary, we know that it has some intercellular clefts that allow for certain things to travel in and out of the cell. When we think about the capillaries that are present at the blood-brain barrier, they're going to have predominantly more tight junctions and minimal, right, of these intercellular clefts. So for the most part, adjacent cells will be completely bound to each other by tight junctions versus allowing varying solutes to travel through those clefts. Meaning that things that can travel across the border of the epithelial cell will be able to travel into these capillaries, travel in and out. But otherwise, you're not going to have larger solutes that have the ability to travel through those open spaces. So we know that things that are easy to travel in are oxygen, varying ions, and glucose. They'll travel down their concentration gradient through transport proteins, and that's how they're able to access, right? But we also know that it's not an absolute barrier, because other things that are fat soluble, like alcohol, nicotine, and varying anesthetics, will be able to pass through the plasma membrane. And so this becomes problematic because things like alcohol and nicotine, right, can have negative effects on those brain structures and create inhibitions, create dysfunctions, especially if used in abundance, and actually lead to full dysfunction or death of a particular neuron, because we know that neurons do not regenerate themselves. We also know that in times of stress, right, or in times of, when we're being stressed predominantly, but then also when we are kind of preoccupied, the blood-brain barrier can be a little bit faulty. So we know individuals, when they're sick and the immune system is kind of being overworked, maybe they're not sleeping as well, those are times where that kind of low permeability and those tight junctions can fail, and then it can allow other things to pass over. So that's why we're a little bit more vulnerable in times of stress, in times of illness, in times of poor sleep, because it actually creates structural changes to our blood-brain barrier. All right, we're going to end there. So we got through the cerebrum, we got through protection of the brain. We'll pick up on Monday with the spinal cord, and then that will wrap up the central nervous system. And then from there, we'll be able to head into the peripheral nervous system.